little green
Or-
thing, right? That glass thing,
Yes,
right?
yes.
And then the one that was sticking up is the,
It's just the extensions of that to actually receive messages.
which is that?
Yeah, yeah, basically,
Okay,
okay.
like that, it's coming out. Like,
By the circle,
ah, okay.
because the other branches are different.
Uh-huh,
Does it look a little bright up here?
yes.
That's what I was thinking, trying to figure out why. So it's so bright there. This doesn't, oh, that's the one we want. Sorry, I'm trying to figure out which one you needed to shut down so that you have more visibility. So basically, one of the things that we do also see, the axon may look more like this. Same type of neuron. You see it, it's one of those things. Now, one of the things that we see is at the cell body, we have what's called the hillock. And the hillock is basically that point where the cell body and the axon come together. And one of the things that we see is you get the message coming from the dendrite to the cell body, and if it gets enough of the message to the hillock, it will send the message down the axon. Okay? Until it gets to the end of the axon, where it can actually release the neurotransmitter. Now, that is referred to, the impulse will actually have what's called the trigger zone. Now, if it doesn't have, and I want you to realize, a lot of the neurons, whether we're looking at it this way or other ways, one of the things that we have is what's called an all or nothing response. You guys have any idea what that means?
It has to be complete, and if it's not, it won't happen.
Yeah, kind of.
Like if it doesn't hit a threshold, it won't happen at all.
That's correct. That sounds a little bit better, okay, for understanding.
If it
So-
doesn't hit a certain threshold, it won't happen.
So like a transformer on a phone pole, it doesn't send out the signal far enough that it won't reach.
It won't get there, no. Okay? And I want you to realize that that threshold has to be met and exceeded to actually send out that message. You can have a message going, a signal going, but if it's not enough, you're not going to get a response. It's not going to trigger the axon to get the stimulation. One of the things we're going to do at some point is we're going to actually look at sensory. I want you to ask yourselves this question, which can be, asked many different ways. But if we actually look at it from a response, one of the things you'll notice is that you can actually have something touching your skin. You can have two things touching your skin. But if the signal at the distance is triggering only one response, it may be normal or may not be normal depending on location. Hands, you expect that it's closer together because of what we use our hands for tactile responses. As opposed to, let's say, on our shoulder, it may not need to be quite as close, or it may not signal at the same distance because of the number of receptors. So let's talk about it in other ways. We talked about your beverage.
You always speak about my beverage. Go ahead.
Because you're always drinking something, that's why, okay?
Who's drinking all my beverage?
No,
Go ahead.
I'm not picking. I just want you to realize. But taste, you get different triggers. Can you pick out all the different flavors that are in there? Or do you only pick out certain ones?
Maybe some.
Some. And I want you to realize that's all about the thresholds of what you're interpreting. You may actually, and it's just like the neurons, they trigger us to a certain extent, but if you're not getting enough of that response at that time, it's not going to trigger a response to the next neuron. or it's not gonna release the neurotransmitters from the neural end plate.
But is it true, like when you're having something spicy, it's not like a taste, it's more like pain?
Yes,
Oh.
because you only have 4 senses on your tongue as far as taste.
Sour.
Sour, sweet,
Sweet.
bitter,
And salty.
and salty. There is a suspicion of a 5th one. They've done research.
Umami, is that what it is?
Yeah, it's called that. It's called savory. They have all different names for it, but it hasn't really been confirmed.
So what taste does that have?
It's basically this stale, other flavor that is not really clear cut. Okay? Where is it associated? That's another question too. There have been patterns, but then again, it's not clear cut either.
And people said like, depending on where you taste the flavor on your tongue, that's where like you taste the certain
That's
flavor.
yeah, that flavor. Interesting, yeah. But I want you to realize it's all related to those tolerance levels, whether it triggers it to go past that point or not. If it doesn't, think about this. Anybody ever see a switching system? Never mind, don't ignore me on that.
Oh, what
was
system?
that?
I can see it's over the heads as far as trying to determine that, so don't worry about it.
You mean like old phone call operators, or like how they do like?
No,
There's-
I was thinking of it actually, where you can actually have levels where it actually has to be, but ignore it, please, ignore it.
I'm kind of curious now. What a switching? What? A switching level?
You get a thermostat. Historically, they've been made of mercury as a switching system, where they actually have the mercury and it goes, and then that actually triggers the response if it's actually higher or lower.
Yeah.
Do you follow what I'm saying?
Yeah.
But if it's actually not at the level that you put the setting, guess what? It actually turns the system on. Does that make more sense now?
No.
That's what I said. Like I said, I forget it, okay?
Yeah.
One of the things that we do see on the axon, we actually have what's called slow axonal flow and fast axonal flow. What basically this means is you have these tubes that are actually going down the axon. Slow axonal flow will be very slow in comparison to the fast. Wow, isn't that, that's very insightful, isn't it? But in all reality, slow is one directional, down towards the end, from the cell body. Fast is going back and forth. Slow will only go, as it says, 1 to 5 millimeters per day. Okay? And you're traveling substances down to the end, specifically neurotransmitters, or parts of the neurotransmitters. When we actually look at fast, it's going back and forth. So it can go down to the end or it can come back to the cell body. This travels 200 to 400 millimeters per day. So it's much faster, relatively speaking. Okay? One of the reasons it does this is because sometimes it reutilizes some of the components that were not used once it got down to the neural end plate. Okay? So it can actually help to redo it. Okay? One of the other things that's really important about this is pathology. Anybody like tetanus? Anyone want tetanus? No, you don't guys don't want tetanus. Do you guys remember what it was made from? Tetanus? A bacteria.
Like the actual
Yeah,
infection?
Which one?
Oh, no, I said the actual
Which one?
infection.
No. You have Clostridium. Clostridium, or tetani. And Clostridium tetani is basically introduced into the bloodstream, and what happens is when it creates a toxin. The toxin will actually be a neurotoxin. Okay, it will cause muscular spasms. Okay, which causes lockjaw. It doesn't just affect the masseter muscle, but it can also affect other muscles if it's not provided a antitoxin to break it down. How do you get it? You broke the skin, you got it in there. Where does this come from? Soil. Contaminated soil with the bacteria. How do you get it? Normally thought of as stepping on an old rusty nail. But the nail doesn't have to be rusty, it doesn't have to be even a nail.
That's every 10 years.
And the tetanus shot to prevent this,
Right.
or shall I say, to have an effect to actually, prevent this from actually having a cause. If you do get a cut, a break in the skin, and it gets introduced, is getting the 10-year tetanus shot.
I recently got
Stings?
it. Stings?
Yeah, stings.
Oh, no. There's pain.
Yeah, it stings.
No, the
A
pain.
lot.
Yeah, because they put it in the
What, the
muscle.
shot?
Yeah, the deltoid muscle.
Yeah.
I know.
It hurts.
Oh yeah.
It lasts.
It's better than getting in the
TMJ.
gluteus muscle. That's all I'll say. But, it does affect muscles, not just the jaw. It can also transfer to other muscles. Okay? So it's not pleasant. And you don't want to drink through a straw, because that's what happens. Okay, and a lot of times if you do get it where you have it, if you get it where it causes lockjaw, you actually have to take muscle relaxers to actually relieve the nerves that are actually triggered and actually having this disruption to the neuron. That's then affecting these muscles. Okay, so this is one of those things that normally has to also occur. We do actually have more than one type of neuron, by the way, as far as how their structures are. Okay, so I do want you to realize there are three basic types: multipolar, biopolar, and unipolar. Okay? So when we're looking at them, the first one, this one right here, this is multipolar. You have dendrites, you have one axon. This is most of your neurons. Bipolar, you have one dendrite, you have one axon. This is what you normally have in some of your sensory special senses, like your ear, your eye, things like that. Okay? You also have unipolar, where the actual cell body is off to the side, so it looks like one extension that's going right down. Unipolar, these are also sensory. Okay? So when we're looking at it, sensory, we did say, are afferent. They basically receive messages from a receptor someplace in the body. We have the motor, which are efferent, which send messages out to the muscles. And then we have the interneural or association neurons. 90% of the neurons are actually in the body, though. Okay? So what's an inter or associate neuron? It looks like this. And we have two types. Purkinje, which you guys have heard of Purkinje. Hopefully you remember Purkinje fibers. when we were talking about the heart muscle. Okay? And then we have pyramidal cells, which actually have basically many dendrites that go to the cell body and one axon that extends out. They need to have all these different triggers from the different sites, the dendrites, to get that one trigger of the cell body. Otherwise, you don't get a response. Any questions about neurons? Okay, let's talk about neuroglial cells. Neuroglial cells, like I said, basically they are 50 of them for each neuron. That's important to realize. They are smaller than neurons, they're supportive, and if you think about it, a lot of them are actually referred to as like the connective tissue of the brain, and areas like that, because they're actually involved in many of the functions. There are basically 4 found in the central nervous system and 2 found in the peripheral nervous system. They're listed up there. They're astrocytes, oligodendrocytes, microglia, ependymal, Schwann, and satellite cells. Now, one of the things that I want you to realize is that, well, here's the thing. When we look at astrocytes, let's look at that. What's the astro sound like? Stars. Good. Okay, so they are star-shaped. That's basically what they are. They actually form the blood-brain barrier. That's one of the reasons the brain does not actually get blood supply. That's also what protects it from actually getting infections most of the time. Okay? It also helps regulate the potassium levels, and it gives support to the brain. Okay? And there's one right up there. See it? There's a blood vessel. There it is, right there. Okay? Microglia, they're small, right? Micro, small. Okay? Microglia are found near the blood vessels. Okay? They have a phagocytic role. So in other words, if they actually get something that doesn't look right, they phagocytize it to try to eliminate it. Anybody drink a lot? You do?
I need a noun. So is it water?
No.
Alcohol?
Yes. Anybody ever hear that alcohol kills brain cells?
Yes.
Good. I'm glad you did.
Wet brain.
Which ones?
Huh?
Oh
Which-
my gosh.
Which brains?
I said wet. No, I said wet brain, like a syndrome of alcohol, like a condition
Yeah.
where somebody's just fermented enough to
Is
basically
that why like you kind of like forget stuff a lot?
alcohol-induced dementia. Yeah,
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 it does
like-
happen. It does
Yeah,
happen.
because like every time like
it does.
you drink, like you just can't like.
But think about this. I want you to realize, and I want you to understand this, because if you look at a lot of college students, they actually have binge drinking. And I'm talking about the typical college-age students. They go to college, they join a fraternity, and they binge drink. What does that mean? On the weekends, they're sloshed. They forget the weekend because it's gone. By the time they wake up, it's Monday morning.
But you do that when you get older too. Unless you have like,
Yeah, I know.
family.
There are people that do that, and they're usually, that does affect them in other ways. I mean, and a lot of times it develops from when they were in college, that whole behavior. But think about it. If you actually have, let's say, 1 drink or 2 drinks, you're actually considered already beyond the alcohol limits for driving.
Well, depending on what kind of drink it is.
1 beer, 1 glass of wine, 1 cocktail.
That's actually a D&D question. I remember. I have no idea.
And you think about it, that's about 12 ounces of beer, about 7 ounces of wine or 2 ounces of hard liquor.
But you shouldn't even be drinking when you're like you haven't thought of you drinking. Yeah, you shouldn't be driving if you haven't thought.
I know, but the students, they start off Friday night and they drink right through. The level of drinking, this is why I'm pointing this out, because the alcohol levels are so high, that's what actually causes a inability to get the oxygen and the sugars the body requires. And that's what really causes problems with the brain and causes damage to some extent. Because alcohol does kill cells. Also, anybody ever have a hangover? Okay, we know about you. You've been drinking. We know, but it's mostly water. Okay,
Yeah,
right?
right.
But think about it. When you get a hangover, what is it caused by? Alcohol. But what is it? It's basically dehydrated. Now, one of the things that happens, and I've seen people do this, they sometimes decide to have, let's say, their cocktail with mixing it with Gatorade. It gives you the potassium
I mean,
to give you those electrolytes, which then retains the water, so you don't actually lose those electrolytes in your brain. Okay, it happens. And I told you guys, when I was young, I did all kinds of wild things. I drank a half a bottle of vodka, just frozen, or in the freezer, straight up like that. Half a bottle, one night. Half a bottle. I'm talking about,
A handle or whatever.
and it was absolute vodka.
ew.
Nothing in it.
I don't know.
Okay? I remember my first time I had tequila, I had the worm. First
Nice.
time. Anybody ever have the worm?
Yep. Gives you a nice little afterbuzz too.
Yeah. I didn't feel it at all.
Yeah.
I have a question.
Forgot.
I took too much.
It's like, dude.
You know how you said that alcohol makes you dehydrated, and that's where you get your hangover.
Yeah.
But like, what happens if you just can't drink anything?
What do you mean?
Because like, if like you feel dehydrated, you try to drink water, you try to drink Gatorade right after the hangover, but like it wouldn't take it.
You mean it comes right up?
Right back out.
Yeah. Well, that's what happens sometimes. Volume is still volume.
And
You
then
can
the
only
acid
retain a
comes out.
certain amount.
And then the
And,
acid.
and I know sometimes that happens. So what occurs is, if it does come right up, guess what? It's not going in and it's not affecting the brain as much.
Oh. So that's a good sign.
So it's a good thing, and you're rejecting it. Your body is rejecting it before it actually can get absorbed.
But what if the acid comes out?
There goes the teeth.
I mean, other than the teeth.
Yep.
What points is it considered alcohol poisoning? Like what determines?
When it actually has an adverse effect, meaning you're actually getting a reaction from drinking the alcohol. I used to know somebody that actually had alcohol poisoning whenever they drank. That means whenever they drank. Okay? But what they would do, if they did drink, you know what they did? Hair of the wolf. You know what that means? To get, or hair of the dog, whatever it's called. I think they went to the wolf, because what they did to get over the hangover,
Drink more.
they would drink more.
Yeah. You can't
And
get
I
alcohol
mean-
if you're drunk.
A lot more.
Right, that's why like if you have a hangover, like the next day, you would have to have alcohol to get it down. Some people do that.
Yeah. But they would actually do this, when they drank, they would actually drink and drink and drink and get more drinks to get rid of the hangover.
So any inebriation is considered like
Yeah.
poisoning.
Yeah.
So it has.
But it's not just they would actually have a reaction to the skin and other
In
things.
what way would you say a reaction? So.
Meaning, like an allergic
And I
type.
have allergic
Yeah.
reaction.
So
So what is alcohol poisoning?
it's when you're getting a reaction, immunological reaction to the alcohol.
As in what?
Like
As in your
you
body
get a
is all
rash,
shut down.
it's actually getting, swelling from it, things like that.
Your body temperature goes down.
So it doesn't necessarily have to do with like the damage, like
No.
the effect of neurons of brain cells.
Yeah, it's
Oh,
other, okay. But a lot of times, and I want you to realize, these cells are going to, once the cells are damaged, these microglia will actually try to clean it up.
Also, they're the good guys.
Yeah, somewhat. Because anything that's debris, that's not supposed to be there, they're trying to clean it up and get rid of it. Okay? So they work with white blood cells in that sense. Epididymal cells, these are interesting, guys. They're actually in certain parts of the brain, the inner portion of the brain, I'll say it that way, which is where we actually produce our cerebral spinal fluid. They're responsible for producing the cerebral spinal fluid. Now, why is cerebral spinal fluid so important? It actually goes into those cavities that surround the brain and the spinal cord. We're going to talk about it more, but I want you to understand. The cerebral spinal fluid allows that flow around the brain and the spinal cord to transport the nutrients.
So when you do a lumbar puncture, that's what you're testing is the CSF?
Not necessarily. When we're actually doing a spinal tap, we are.
Spinal tap, okay.
Yeah. Spinal tap, we actually use it to do a couple things. We test it for pressure, which shouldn't be too much pressure. If it squirts out, it's too much pressure. That means there's an infection.
Meningitis.
Meningitis, typically. What we can also do is culture that to check what's going on. We check it for, what's going on as far as if we can actually get movement, flow of it. And that's important too, because sometimes it doesn't have the flow it's supposed to. Okay? So there's a couple things we can actually check with it. We can also introduce medications there, like, you guys know about epidurals?
For birthing? Yeah.
Well, epidurals are not just for birthing,
Or just
for pain management also.
You gotta be careful with that.
Yes, they do. And you look at it, they're actually introducing the needle along the spinal cord in the space to introduce the medication.
That's why if you have scoliosis, you have to tell them ahead of time.
Yep. Okay. When I had surgery, I had 2 anesthesiologists working on it to make sure they got it right for an epidural for the surgery.
It's like if they hit you in the wrong spot.
Yep, you could actually have serious problems, including paralysis possibly. Okay. So cerebral spinal fluid is really important to understand, and it should be clear like blood plasma. And free of bacteria or any other microbes. So it should be sterile under normal circumstances. But the epididymal cells are what produces this. And it actually goes into the 4 cavities that we have within the brain. And we're going to talk about them. We actually have we'll talk about the 2 lateral cavities and then the 3rd ventricle and the 4th ventricle. Okay? Which then gets us to what's called satellite cells. One of the interesting things that we see, satellite cells, they are found in the basically in the central nervous system, but they're actually very similar to Schwann cells. What they do is they create myelin. Okay? That's basically what they do. Okay? Oh, excuse me, not myelin. these, I'm thinking of oligodendrocytes, my mistake. These aren't peripheral, but they're actually supportive of the, in the peripheral nervous system. Oligodendrocytes and Schwann cells basically produce myelin around the neurons, specifically the axon. One of the interesting things about myelin that I want you to realize about this, I want you to think about this in this sense. Let's get a neuron. Dendrite, cell body, axon. See that nice axon? Okay, nice axon. Another cell body, very similar, right? About the same length. Okay, you guys see it? Okay, when we're actually looking at the transmission of a neuron as far as its stimulation, we're actually looking at the exchange of sodium going in because of permeability. Okay? You guys follow that? So basically it's the permeability of the sodium rushing in if there is enough at the helix to create the axon stimulation. Well, a lot of times, if it's, and I want you to see this, if it's myelinated, what happens is the myelin, which is either the oligodendrocytes or the Schwann cells, oligodendrocytes central, Schwann cells peripheral. What we see is this. We actually have the cells going along that axon. Now, when we actually see the triggers, we'll get it triggered here, peripherate, it gets more peripheral. Once it goes, and it can go here, and has that mechanism going. But when we actually have it going here, what happens is it skips over where the Schwann cell or the oligodendrocyte. Why? Because it doesn't have access to the periphery, the surroundings of the membrane.
Okay,
So it skips over.
So like it would skip around?
Yep, and then the next one skips here. The next one, I'll put another line like that, skips there. The next one, over to here. The next one, over to here. So the next one, over here. So what happens is, in a myelinated neuron, it has a faster transmission of the signal. Because it skips over where the myelin is located.
Right.
The location where it actually has these breaks, where it can actually do this, are known as the nodes of Ranvier. Okay, you want the English pronunciation? Ranvier. Okay, so the nodes of Ranvier are the location where we actually skip, where we actually have access to the outside, where it actually can get the sodium rushing in when the cell becomes permeable.
Oh, so like,
Yep.
like the spaces in between?
Yeah, in between each
That's where the
cell.
sodiums will go in?
Yes.
So the Randier can skip over the myelin? Or not, has nothing to do with it?
No, that's the spaces where it actually has access, okay, to the surrounding tissue. Okay? So when we're looking at myelin, it's basically made of these cells, and they surround it, and they actually, allow for this electrical impulse to get faster because it's actually skipping over that portion. Okay?
So that's protein?
Yeah, well, it's got a lot of protein because it has a lot of lipids because it has a lot of membrane.
Also, it's lipid and protein?
Yes.
Oh, okay.
So when we're actually looking at it, here's the typical Schwann cell. They actually surround the actual axon.
So is that yellow part?
That's the membrane.
The lipid? Okay.
That's the membrane of
Well,
the cell.
well, like in the middle, yeah, that is. But like.
So what you're seeing is right here. I mean, that's what the overall impression is, but if you're looking at here,
Right.
this is the cytoplasm, here's the nucleus, and basically all this is membrane that's actually overlapping.
Then where's the lipid and where's the
The lipid would be part of the actual membrane, because membranes have, they have the phospholipid bilayer, and they actually have proteins within the membrane.
Oh, so it would
That's
be in that yellow area?
Yes.
Okay.
Okay, so when we're looking at it, it could look like this. Notice in this, we actually see the node of rhombiate towards the middle of those two cells that are surrounding the axon. And as they described it, it looks like a jelly roll. Anybody ever have a jelly roll? A jelly roll. You never had a jelly roll?
Delicious.
You don't know what a jelly roll is?
What?
Oh my God, oh, it's a cake that actually has jelly in between and they roll it, so it actually looks like that basically. That's all. You never had a jelly roll?
It's one of my favorites.
What's that?
It's one of my favorites.
Yeah, you know,
It's
it's a
delicious.
jelly roll. Who hasn't had a jelly roll? It's jelly where the cake is actually wrapped around with the jelly on. You got
Oh, like those custard looking.
No, not a custard. No.
He's literally saying that there's jelly.
Yeah, there's jelly.
It's
They
literally
cover-
a cake that's rolled with
And
jelly.
they cover it with jelly and then they roll
Just
it.
look it up.
Yeah, look up a jelly roll.
It's amazing. It's
And then they cut
simplistically
the whole
amazing.
thing where you see it look like a, you know what I'm talking about? Yes. Do you know what I'm talking about? Oh my goodness, you have to join him on this one.
Professor?
Yeah.
Never mind.
What?
Yeah, it looks like custard.
This is what I see.
You know, I mean, you might as well be looking up Fat Joe.
What?
Oh.
You know who that is?
Yeah.
A rapper
Where do you see jelly in it?
from New York.
Oh, just that?
Nah, you lost me
You
on
know
that
who I'm
one.
talking about.
You lost me on that
It's just
one.
like
She knows who I'm
a,
talking
like a
about.
fake custard.
Like if you cut it off, it's.
I mean, the pictures she got look almost like Fat Joe.
I mean, honestly, look up
Yeah,
jelly
fact.
roll, you're gonna see a rapper. Wow. What would I know, right?
I'm just, you know.
Just look up jelly
Goodness. I don't know.
Jeez.
Okay. Okay, so we look at that. Myelination, by the way, is what actually changes white matter to gray matter. White matter has myelination, gray matter does not. Okay?
So pretty much like the inner.
Yep, of the brain.
Okay.
Also in the spinal cord. The outer portion of the spinal cord actually has white matter, as you see on the left. That's the spinal cord. Okay? So when we're looking at it, white matter is myelinated, gray matter is not myelinated.
Which means.
So gray matter has a slower processing time. The periphery of the brain does not, has gray matter. That's where you get your deep thoughts, your contemplations, things like that.
Yeah, the gray matter?
What's that?
You said those are the gray matter?
That's the gray matter. Okay? Whereas the, how do I say, the more homeostatic aspects is in the white matter. Where you get the responses, things like that. For instance, reflexes. White matter. Think of it this way, if you go out tonight and you start driving down the road and a deer runs across the road,
Oh my god, that's my biggest fear. I always think about that. I'm not
it
even
happens.
kidding.
It happens, believe me, it
I
does.
know.
I knew somebody that hit a deer.
Oh my gosh, what happened to the
It-
car?
The car got the.
Yeah, they got rid of, the whole front end was damaged.
Insurance don't cover that, right?
No,
No.
no, because it's not an accident caused by another vehicle.
It depends on the insurance I heard.
Mm.
There is such thing as
Acts of
deer
It's not
coverage or
including
whatever.
full coverage?
No, there's a thing called deer coverage or whatever. it
Deer
is.
coverage.
No, there's such thing, like, you gotta look it up.
I guess you have to have deer coverage.
No, there is a thing where they cover it for you, but you have to have that in your plan for it
Yeah.
to be covered.
It's sort of like, when you actually have flood insurance. You have to have a flood zone, and then you're paying deep pockets. Deep pockets. We almost bought a house where there was, in the garage, it was designated as a flood zone, so it actually covers the whole property, so you have to get flood insurance for the whole property.
Oh my
We decided not to buy that.
God. Yeah. Especially when the
Because
garage
it,
go
like-
down, it's not gonna.
No, it really increased the price of the, insurance
Right.
considerably. Like,
Right.
almost the price of the mortgage itself.
So the
Yeah,
CS,
it's like property, yeah, property is very, yeah.
We're talking about substantial, just because it was in a flood zone.
so CSF has an encapsulation around the brain and the spinal,
Mm-hmm. Yep.
just on the outer
Yes,
bit?
on the outside.
So
That's
there's no
actually those membranes that we talked about, the meninges.
meninges.
We're gonna call it that way. It's the meninges. In the past, you've heard of it as being the vertebral
Okay.
cavity and the cranial cavity. Okay? But now we're changing that because now we're being more specific to this portion of the body. Okay? A couple things we need to also talk about. We have neurons, they do have electrical signals. They do have action potential and they have graded potentials. Basically, what happens is both are occurring. Okay? Action potential is basically where it travels long distances. Okay? Like on the axon. Graded potentials are where we have it localized, where we actually have, let's say, an electrical occurrence at the end of the axon to actually cause a stimulation to release the neurotransmitters. Okay? So that's where we may see it. We also have, one of the things that happens, potassium leaks out continuously. So that's one of the reasons. Potassium is also very important inside the neuron. Sodium will rush in when we actually have a leakage channel, or what happens is the channel opens up and we actually have it leaking right in because it's going to rush in. Typically, we actually have the differences of being a negative 70 millivolts. That's because the sodium is higher on the outside than the potassium that's on the inside. When it actually has an action potential, what occurs is the sodium rushes in, the potassium still keeps leaking out, but the potassium helps as far as the recovery back to its resting state. The gated channels will actually get excited though, which is what we see at the synapses. The ion channels, one of the things that we actually do see with them, we can actually have voltage gated channels, which is basically related to the change in charge. We have ligand gated channels, which is basically because of a neurotransmitter that is actually coming into that channel, or and it's actually, or shall I say, could also have, let's say, something like calcium coming into the channel, which will then open up that channel. A lot of times it's actually the sodium, because the sodium will actually go into the membrane and actually open it up for another location nearby. We also have mechanical gated channels. This is where you actually may have a response because of some sort of a vibration, a mechanical occurrence. Let's say a sharp object hitting nearby and it triggers it. Okay? So when we're looking at it, we have all these different types of channels that could actually trigger a neuron. So here's examples visually. Okay. A lot of times the channels will have a resting potential in the membrane, and like I said, A lot of times it actually will have the sodium higher on the outside than on the inside. Potassium's higher on the inside of the cell. And what exactly happens is you have a permeability. The permeability allows sodium to rush in, and what happens is the sodium then gets, where you have a voltage change, a current occurring, basically. Okay? Look at this diagram. You'll notice the distribution of in this case, on the left, you see the positive and the negative. The positive are green, the negative are red. Okay? If you count them, you'll see more green on the outside than on the inside. On the right, you see chlorine and sodium. Sodium is green, chlorine is yellow, phosphate is blue, protein is red and it's big, but you see a difference from one side of the membrane to the other. And it's this gradient that actually you might see differences and changes. So that when permeability occurs, electrolytes will move through. So when we actually have a gradient potential, basically what happens is we have a signal. We have the resting, and it actually has the resting membrane where it actually gets an impulse, a depolarization event, and then it actually gets repolarized. And it goes back to resting. So how do they come about? You get vibrations. You get chemical stimulations, like ligand glutathione, a neurotransmitter basically affected. Okay. By the way, neurotransmitters, some of the neurotransmitters you might come in contact with actually resemble or have the same names of certain medication or certain things that we give people as medications. For instance, you might notice, we'll talk about them in a little bit, but I want you to realize, and some of them will actually inhibit, some of them will actually affect both of the membranes across that synapse. So we actually have a presynaptic membrane and a postsynaptic membrane. The presynaptic membrane is on the axon, which releases the neurotransmitter, which may increase or decrease the neurotransmitter that's released. The postsynaptic membrane is where we actually have these receptor sites, where we can actually have the ligand channel. And basically what happens is you may block the channel, or you may actually have increases or decreases to the channel of the postsynaptic membrane. When we actually look at it, some medications that are given to patients, like we talked about alcohol, is a depressant. So it depresses generally the nervous system, even though people get more thought that they are less inhibited, but it depresses it overall. We have caffeine and other things that may stimulate, and some will actually do a little bit of a flexing of it. So I want you to realize that. Okay? We have the graded channels, which are basically ions that are affecting it, like calcium. Ions. A lot of times when we have action potentials or the impulse, basically we start off with a resting state. It's depolarized because basically the sodium rushes in, changes the charge, and then we have repolarization before we get back to resting again. And like I said, we either have, it's an all or nothing type occurrence. You either get a response or you don't get a response. If there's enough stimulus, you get a response. If it's not quite that level of the threshold, you don't get the response. You don't even know it happened. Visually, graphically, this is how it looks. You have the action potential, the stimulus, you have depolarization, repolarization, you also have hyperpolarization that occurs, which is excessive amounts of the potassium actually occurring, and the sodium potassium pump is also forcing the sodium out, more so than it's needed, and then it actually settles off. So that's basically all these are really going over those details. when we actually look at the action potential, one of the things that we see is it also affects these channels, okay? So let's talk about anesthesia. Guys like anesthesia? Ooh.
Well, I've never had them.
Never had it?
That's
Yeah.
a question, if
Yes.
you can possibly put this in perspective. So what's happening, generally speaking, when individuals given, say, like two individuals are given like Adderall and one's more stimulated while the other one's depressed?
A lot of times it may be effects from other things that are happening also. Remember what we're looking at, a lot of times we have variations in the individual, okay, as far as their specific chemistry.
So it wouldn't necessarily be like a deformity in like the
It
synapses?
depends.
Oh.
It depends. I had a student that actually, what happened was they changed her medication. It actually threw her off that she had to get lithium to actually get stabilized, and it took her about 3 months before she actually came back to normal. Her friend told me because she had to drop the class because of it. Okay? Exactly. Local anesthesias, like Novocaine, lidocaine. Anybody ever see those? Anybody use those?
Yeah.
Okay. Basically, it's a localized. It blocks. It basically it opens. It blocks. It blocks the voltage gated channel for sodium. So it goes in there. It blocks it. It prevents it from occurring. So what happens is you don't get the effects of the stimulation.
For instance, the lidocaine, the patches, and kind of like
Yep.
gum shooting.
Yeah. Or you actually have rubs, you know, these salves that they actually use. But, you know, lidocaine is more topical. Novocaine, they actually use it in anesthesia to actually, or for dental work a lot of times, to actually numb it. Yeah. When we're actually looking at the action potential, basically it's related to sodium actually doing this, traveling. And it can actually travel over an area. Now, you do actually have what's called continuous versus solitary. Basically, continuous means it's basically a continuous step by step, like what we were talking about over there. Solitary is where it actually goes over the nodes of Brombier. So our two examples, the top one is more continuous, where there's no myelin. The bottom one is where it's solitary, where it skips over the myelin cells. And we see it in this picture. Okay? It also, as we talked about, the solitary has a higher speed for that transmission. I'm going to mention that there are 3 types of fibers. I don't want you to worry about them. They're A, they're B, and they're C. A fibers are the largest, B fibers are medium-sized, and C fibers are short. We will hear it again, A, B, and C. I will tell you that much. A, we actually have it on somatic, they're myelinated. B, we see it on myelinated, visceral, and autonomic, preganglionic cells. C are the smallest, they're unmyelinated. Okay? You just listen to it, you heard it, we'll hear it again. I just want you to understand when you're actually hearing it again, what they are, that's all, okay? Stimulus. When we get a stimulus, it can have 2 things that affect it. How often, and the number of neurons that are actually affected. Anybody ever get pain? You guys like pain?
No.
Do you want to get rid of pain? Do you want it never to come about?
I mean, not necessarily. I feel pain when I eat spicy food. I like that.
Yeah. Pain is beneficial. You know why?
Because it
Pain
releases
is a good
a-
indicator of something that went wrong. There are people that have no perception of pain. And when that happens, they don't know when something is off.
What's that chemical that's released after experiencing something like eating something spicy. It's begins with a C. Cadis? No.
Capsaicin.
Capsaicin.
Okay. So when you're actually looking in stimuli, it's related to how frequently you're getting the stimulus and how many neurons are affected, basically, to get that stimulus. Let's go on to synapse. Two types of synapses, electrical and chemical. Okay? When we look at a synapse, it looks like this. You guys looked at this, the motor end plate, okay?
That was the ball that we
Yep.
saw, right?
Yep.
With the little balls on the
Yep,
bottom.
that was the balls. Those are vacuoles. Inside the vacuoles are the neurotransmitters,
Kinda looks like a bell.
okay? The calcium channels will actually stimulate the vacuoles with the neurotransmitters to come close to the membrane and attach to it for phagocytic occurrences, specifically exocytosis. Okay, so they can release the neurotransmitter if they are stimulated. Okay, to do this, that means the signal has to stimulate them, and they have to have calcium ions that with the voltage gated channel to actually cause it to function. For the presynaptic membrane, which is the axon bulb. Okay, if the neurotransmitters get into that synapse, they undergo diffusion. They just simply diffuse across from the area of high concentration where they release to the area across from on the postsynaptic membrane of the dendrite. Okay? And a lot of times the neurotransmitter attaches to a ligand channel and it opens up the channel, which then allows sodium to rush in, causing the stimulus. And like I said, synapses are one-directional also, because the postsynaptic membrane has the receptors. The presynaptic membrane is what releases the neurotransmitters. We do have inhibitory and excitatory potentials. So a lot of times it depends on whether it's exciting or actually inhibiting the response. So you'll see EPSP at times, which is excitatory, and IPSP meaning inhibitory. Okay? So what do we do when we do have a neurotransmitter? We can actually have it diffuse across. That's how we get rid of it. We can have it broken down with enzymes like we did with acetylcholine, broken down by acetylcholine esterase. We can also have uptake inhibitors, like Prozac is an reuptake inhibitor. So it prevents it from actually doing its job. Okay. Three responses you have are basically excitatory occurrences. You have a threshold that is reached, or you can have inhibition. And a lot of times you have graded and action potentials happening, which will be related to how much of the response is going continuously, so all or nothing wise. How long it's lasting, the stimulus, and how long is the refractory period where it actually is able to recover from it. Which gives us the summations. Summations could be spatial or temporal. Spatial is we get a signal that actually is a spike, basically. And this is where it actually exceeds the threshold, getting one response. Temporal is where we actually get it, where it's one neuron affecting the next neuron, basically. Spatial, you can have 2 neurons affecting the one. Okay. Overall, we're looking at whether it's getting more excited or more inhibited responses. Okay, overall. Neurotransmitters, excitatory, inhibitory, to both systems, central or peripheral. And we have a bunch of them. So why don't we stop at neurotransmitters? Okay? This
Can you
way-
please publish this? I can't write that fast for some of these conversations.
Like I said, this is why I go to PowerPoint because it's so much detail.
Yeah, can we have this on Moodle?
I'll see what I can do. All right? There's a lot of details, as you can see. I only have 16, 17 more slides on this particular one.
On this
But the
chapter.
brain is broken